sheet that I gave out for the midterm is here. If you don't have one from before, it's also on Moodle in Bowles week 6. And this week, questions from the various units that we've discussed. And I see some assignments in the basket. If you have them anytime, send it to Moodle or bring it by Monday after the exam Monday, and I grade them, and that's the end. So the deadline is definitely Monday by the exam. And that leaves us with your questions. I hear a lot of hype about blue code.
I'm pretty sure that's
The only answer to every question. That's kind of like choosing C on multiple choice. And give it a shot.
Stand to it.
it's gonna be right
Yeah,
at that one
gotta
point.
see that bit through. Sort of break the ice here. Maybe, Julie, you want to ask a question from the class Monday?
Yeah, I was basically asking like what the actual difference was between an e-mobile and a mobile nutrient, like what that actually meant and why one wasn't mobile and one was.
Yeah, so we were getting to discussion about mobile and immobile, and this was, like I said, kind of the crux of the plant nutrition situation. Again, the mobile nutrients, we're talking about mobile in the plant, plant mobile nutrients. We see deficiency symptoms in the older leaves first, plant immobile nutrients in the newer leaves first, and Julian's question is why? Why can't we move them around? Right? And part of the answer is that it's different for each one. So there's, in each case, for calcium and the metals, there's something about size and shape once they're incorporated in, so it's a connection between those things and where it's used, like which molecules, which structures it goes into, that makes it so that the plant can't break that back down and then move it again in phloem. So those things all get to that location in the new growth, dissolved in water, like I've been talking about, right? That's how things get into the root in the first place. And so if it's dissolved in water, then what transports it to that new growth? The xylem, the stuff that moves water. Yeah. So oftentimes it can be moved in solution, but then when it's taken out of solution, that means dissolved in water, and incorporated into the structure, now it can't be broken back down because of its size and moved through phloem. Because xylem's only going up with transpirational pull, right? And so that's usually the limitation, is that it can't actually be moved somewhere else. Whereas because of the shape and the structure of these other molecules, they are able to be moved up from old to new in xylem. Right? But these big things can't necessarily move down in phloem. Does that make sense so far?
I think so.
The look says no.
I'm just still thinking about it.
What's that?
So like the key difference is that the mobile nutrients don't get like broken down and incorporated into the structure of the plant the same way that the mobile ones would. So that you're saying if like water is introduced again, it can move, or is it just purely the structure difference?
Okay, right, so this is just like that slide that I used. Here's new, right? Here is older. Whatever it is, it's coming in. Let's get this thing roots. was just going there, right? And now, these leaves are getting formed, water is moving everything there. But now that it's in structure at that point, it can't be moved back down in flow. On the other hand, those nutrients that are here, right, and the magnesium are getting pulled into the xylem and up. And the reason why we can't take calcium, say, from this spot and go up is because of that size and shape.
So it goes up the xylem, and then for the mobile nutrients, it goes down the flow. Because they all rise up in water, and then, so that goes through the xylem. And then for the mobile nutrients, when they're going back down, because they're small enough, they can get through the flow.
Well, when the mobile nutrients are being mobilized to the new growth, they're still going up, right? It's these going from this location to this one. Right? But what I'm saying is, then in theory, anything that's here could go up, right? But it's something specific about the structure that it's building and the shape and size that it has that makes it hard to do that. And so that's why if it's not immediately put in place here, then you can't get it from elsewhere. And it ends up being that you start building these structures with an incomplete set of materials. And
So we also use mobility to describe nutrients in the soil.
yeah,
Is it the same classification of mobile and mobile in plants and soil? Like nitrogen is mobile in both?
not always.
Okay.
So yes, we use the same words, but that's why I'm trying to be clear about plant mobility versus soil mobility. And so nitrogen is what happens to be one that's mobile in both places. Phosphorus, mobile in the plants, but not in the soil, for example. And so potassium, mobile in the plant, and like marginally mobile in the soil. So there's not a consistent relationship there. And that has to do, instead of with size and shape getting moved around inside this living organism, in the soil, it's about how those things are held on to those soil particles. Claire, do you have a question related to that?
No, that was, I did not know.
You're like, I'm wondering the same things? Yeah. Right, and so anytime you hear that, mobile, immobile, sometimes, like contextually, it's obvious, and other times you want to make sure which thing you're referring to. Am I getting there, Julian? A little bit better?
Yeah, no.
The part that I'm not saying is like the individual chemistry explanation for each one of these. And it's like, it involves a lot of factors. I don't know all of them. I couldn't tell you specifically why zinc doesn't move. But for each one, it's a complex combination of those things.
Yeah, I think I more wanted to understand like the difference between the two, which that definitely helped. And like, I guess I'm thinking now of like the mobile ones, like you kind of have one shot to get that nutrients where it needs to go, because once it, because like it could still get anywhere in the plant through the xylem initially, but then once it gets there, that's in one spot, and if you can get more.
Exactly. Once it gets there for the immobile group,
Yeah.
right, then it's stuck at that location. And so whether that was when this was formed, when this was formed, or now, trying to create this next one, that's it. It stays in that structure until plant dies and it breaks down, maybe it gets eaten, whatever the next step is. But for the mobile nutrients, they've got this ability, and oftentimes you can make a relationship like the ends that exist, the P's and the K's, have, once you break down the P2O5 and the nitrogen, there's these singles, but it doesn't hold true because magnesium has 2 pluses just like calcium, but one is mobile and is not. So there's more pieces to the puzzle, but, this connects to the shape and size. And so those, because of that, can get shuffled around, and that's really useful, right? And that's part of like, when you see, chlorosis in the lower leaves, on the one hand, now you know there's something going on, right, with a mobile nutrient. And two, just as like a plant manager, you know, even your philodendron on the coffee table or whatever, sometimes the initial reaction is like, pluck that thing off, it doesn't look good, right? But give it a chance. It's gonna move all the nutrients it can to that new growth, right, from that leaf, you know, and then once it's fully chlorotic, you know, then take it off of there, no need to let it rot or whatever. But, right, that's material there, that's fertilizer that it can get to the top. Don't chew your lips about that one. Yeah.
Can we go over like the structure of like Winnie, dichots and stuff like that again?
No. Yeah.
Feels so long ago.
Yeah, exactly, that's why that stuff's good to bring up again, right?
I mix up the primary and the secondary xylem, like which one's with it, which one's not, all of it.
So, where do we start, Mel? Monocotyledonous dicot or primary versus secondary?
Yeah, it's really like the Winnie dicot structure, I think that is basically up to
So the core message here, primary xylem is that new functional xylem each year. And the secondary xylem is last year's and then the year before, right, and then the year before that, and it's dropping in efficacy and function as it gets older and older.
and it's also just for like storage. That's
Yes.
sort of every-
So first off, here's in green, vascular cambium, right, which is what we refer to that ring of meristematic tissue, specifically in woody plants, because it exists in the ring, whereas in the herbaceous dicots, it's bundled, right, and they're not necessarily contiguous. Sometimes it can be, but as a rule, in woody plants, it's always continuous. So, meristematic tissue here, one new cell layer to the outside, making phloem, one new cell layer to the inside, making xylem, and that's your functional this year's xylem.
Okay.
Primary was the first one that was laid down, right? And then secondary as it made more and more and more and more.
So, I need a little
Awesome.
clarification based on the first thing, so that's the primary xylem, it's the initial xylem, but it's like every time, each year is gonna be a new primary xylem while the rest are secondary, or the rest are secondary, and only the first one is the primary?
Yeah, so thanks for pointing that out.
Yeah.
I did say it unclearly. The primary means first,
Okay.
right? So that's the first one that was made. And then in its first year, every ring of secondary xylem is active. And then in subsequent year, there's a new active layer, and that one's pushed down.
But the new active layer is secondary?
Yeah, because it's, I mean, except for the very first one,
Yeah.
right? And that's why your primary is as far away as possible from the vascular cambium, because it's been now 2 full years in, and so it's buried closer to the middle of the trunk in that heartwood
So the secondary xylem is the active one?
in its first year.
In its first year?
Yeah.
Okay.
Just this layer here, and then next year, that's gonna be here, okay, and losing function. And then to your other question, the vascular rays, which connect all the way out from the cortex, through the phloem, through the xylem, to the middle, in the pith, are used for
much space and how far it is to get there from it. John?
So with the xylem, it's just that the current year, that's functional, right?
Mostly, yes. There's a gradient of like, this one fully functional, this one losing, yeah, maybe a third, maybe half, you know, and then by the third year, non-functional.
Is that the same for the phloem?
No,
All of those, all the, can the primary phloem still function?
yes.
Okay.
And it's making a much smaller layer
Right.
each year, right? And because it's staying active and it's on the outside, that's possible. Because that xylem on the inside is needed for support. Remember, this is a tree compared to like a smaller herbaceous plant for structural integrity, for standing up tall against gravity. And so the more woody structure on the interior, the more solid it's going to be. At the same time, it's a benefit to not have to spend energy hydrating, feeding all of that interior. So it's advantageous to incrementally, be decommissioned out of xylem function so that you can just be the skeleton that doesn't need food and water.
I wasn't able, sorry, this is a new topic, we're not ready to go on.
Let's see, others on this. I should also come back to it.
Can we just quickly touch base on the court payment?
Sure.
I do not know what I should know.
Oh, yeah. Yeah, I didn't spend a lot of time on core camu. I think all you need to know about that is it's a second layer of meristematic tissue. So that you hear cambium in there, you think the same as vascular cambium. It's a meristematic tissue that makes the cork. It makes the bark to the outside. And by doing that, it protects this phloem so that it doesn't have to reproduce its whole self every time, because it can just keep functioning as that thin layer with protection. So it's like penned in there, and it can stay useful instead of getting damaged from the outside.
So is it creating only tissue to one side, whereas the vascular creates, xylem phloem on either side?
It does that also. It creates that cork to the outside and the cortex to the inside, all of which falls under the bark category.
Thank you.
So for the vascular rays, they can push stuff into the more interior part of the tree. Are those like, can it push it through to dead things, or is it only pushing and storing stuff that's alive?
It's using the non-functional dead xylem for that storage.
It just like shoves it back there?
Yeah. So it's mostly not storage that it's expecting to use later and get access to.
Yeah, it's just
It's
like-
mostly hiding things away. Yeah.
It's more like excess than storage.
Yes. Or in some cases, it's like some sort of toxin from the soil, maybe heavy metals that happen to be there that aren't in the plant micronutrients that I mentioned, right? And then it's going to shove that into dead cells so that it doesn't actively have a damaging negative effect on the living cell.
How does it get there though?
Does it get in there?
They,
Like what moves
like,
it
cause like,
from here to here?
yeah, before we get off of this, I was just curious, like, if you cut into a tree, sometimes you see like pockets of sap, like way in there.
Yeah.
And it's like not, now I'm learning that that's not the living part of the tree, why is there a bunch of sap in there? And like, is it just like randomly like just making little blocks inside of it?
Sometimes, yeah. Like, and the answer varies, you gotta ask the tree, about what happened to it. But sometimes, like that little pitch pocket, right, could be, oh, there was, say, a boring insect, it's gone in, and now it's like, gonna seal it away, right? It's gonna trap it in there, in the amber, and then we can make Jurassic Park. But I mean, but seriously, right? It's just trying to trap that thing. With, again, just like we were just saying about protecting from the heavy metals by not affecting the living tissue, protecting from the insect damage by stopping that thing from continuing through. And that's not the only explanation, but it's an
Yeah,
example
but it's
of how
just
that
like
can be used.
pushing it as far as it can with the living tissue into the dead stuff.
Yeah.
Like, that's where I'm.
Others here? And like I said, we can also come back to it, and I'm sure a question later will then connect to stems again. Definitely be some questions about stems, especially the woody stems, though not as many as on midterm. Related here, and a couple different ways to look at it. Again, back to Sal's question, primary xylem as far away as can be, second, third, right, et cetera, up toward that living vascular cambium meristematic tissue. Dicot stem, what's in there, how that's different from a monocot stem. Maybe a question about that. Remember, secondary growth is just in dicots. The monocots don't have vascular cambium. They have these intercalary meristems instead. We talked about the parts here, like the sapwood is functional xylem, hardwood is non-functional, dead xylem now, where we're gonna hide those things away for later. We talked about gymnosperms having tracheids and angiosperms having vessels and tracheids, moving water faster in the spring, right, in these big tubes, with this risk then of embolism or air bubble, because it takes more water to fill a wider straw.
The tracheids are the smaller ones, right?
That's right.
That's smaller, yeah.
Yep.
Understood.
Vessels move more, and those are only found in the angiosperm species.
Right.
Woody species. Yeah. Claire, to your question.
Actually, well, I guess like the phytochrome period, phytochrome conversion cycle between PR and PFR. Because I missed that day,
Yeah.
did you go more in depth than what's in this couple of slides?
No,
Okay.
just the core takeaway here is plants sense the dark period. So even though we say short day and night, long day plant, it's the actual measurement is of darkness. First concept. The second is the difference in the speed of conversion between these two phytochromes. The far red is converted slowly to red at night, right? And that reverse is converted quickly. And that's what, so red to far red is fast, and that's what that flash of light, short exposure, has the capacity to quickly turn that all into far red, saying it's daytime, even if it was just a couple minutes. And that's how that long night or short day plant gets the night split in two and interpreted as two short nights. And then with prolonged history of that can delay the flowering. Or on the flip side, in the long day plant, it would induce flowering. So,
Yeah, so this far red light, it does convert it back to PR then?
it does.
Okay, cool. I just want to make sure I get that right.
So, slowly over the night period, right? So on a longer night, the plant senses that there's very little PR, and that says long night. On a short night, the plant observes more PFR, and that means short night.
Got it.
Because it's about how quickly this thing breaks down, which is slow. And this one, anytime there's light, it's been very rapidly converted.
Gotcha.
Yeah.
Could you go over far red?
Yeah.
Like,
And the big thing here, go back to my visible light spectrum, is, so again, red light's here, far red is here, just into the infrared spectrum. difference between daytime red light that's useful for plant growth, that hits the chlorophyll peak on this side. So you can see we're calling it red, but it's kind of orange red. But you see then, by the time it gets to red, at 700 nanometers, it drops off, right? And so this far red light, it's not energetically useful in photosynthesis because, in part, it shows up at sunset as the energy of the sun is waning. And so, that quantity of far red says the amount of time without light, the darkness. Whereas that red light, because it is energetically useful, when it's absorbed, it's quickly converted away. And so it's measuring how much of that time is post-sunset, basically, until more light shows up and that all disappears.
Okay, so the PFR is kind of like a stopwatch.
Yes. It's like a very slow stopwatch. Or like, I think Dee said it the first day I was talking about it, like an hourglass.
Okay. Yeah, that makes
And
sense.
when you flip the hourglass, when light hits it, this one flips over fast. And when it's here, then it trickles through slowly. And how long it takes to get to the bottom, you know, how much is in the bottom by the time the sun comes up, says, was that a long or a short night? And then the sun comes up and it flips over and it starts going again.
That makes sense. Thank you.
The sands of time. Yes. Others about that?
Well, this is something different. Can you remind me what the word was for when water moves along the cells, not through?
Apoplastic.
It's in my
That will
notes.
not be on the exam. But the role of the Casparian strip, you gotta get right across this thing and go into the cell. Definitely still very good.
Getting into the Casparian strip is always active transport.
No, but it's always symplastic. It can still move across that membrane via osmosis if the gradient's correct. But it has to be inside the cell. Before I go there, Claire, is this, help?
Yes, that's right, thank you.
That gives us way back in roots, I believe. Again, apoplast, technically not in the living cell, but between the cell wall and the cell membrane. It's easier. You don't have to keep going across. You don't have to consider the gradient. Right? Easy to do. A lot of nutrients are moved that way because of their size. Much easier than pushing them across, making sure the right membrane protein's present. But you get to this barricade now, right? My bouncer analogy, and you got to go inside to get around them. You got to get in the club to get around the bouncer to get over into that xylem vascular tissue. Gotcha.
For the pericycle is the beginning of meristematic tissue, right?
Yeah.
And it's connected to the vascular system or is part of the vascular system?
It's a layer of meristematic tissue that it's not vascular and that it doesn't move those things, but it's there for
Yes, that.
the establishment of new roots. So that's where those are formed, and then that root is both connected to the xylem, which is like the next cell layer over to the interior, and connected to its role as a root, doing water absorption through those root hairs.
So it's like a connecting point of like the endodermis to the vascular system?
It's like, it has that role, but it's also meristematic.
Right.
Could we go over like the different types of fruits?
Also roots.
Yeah.
Can we also go over with the roots,
structures, how many flowers and how many ovaries. And so that's, for this, just know what a simple fruit is, an aggregate, and multiple. But I'm not gonna tell you, ask me like, tell me if peach is a drupe or a samara or whatever. That can be for a different class. So, the consideration is fruit type, number of flowers, you gotta read it. Symbol has a clue in the name. Simple, few, not complicated. One flower, one ovary. So this, all one big ovary, right, with the seed inside. And we do call that situation a drupe, and they all have the qualities of what's inside the seed that we've talked about. Encasement in some kind of pericarp that's fleshy. But in the others, it's like multiple units of this put together. Right? So then that complexity builds from symbols. These are still symbols. These are berries. Okay. Also simple. Still. The apple, I think, is useful because you can see, here's where the flower was, one flower, one ovary. Multiple seeds, multiple ovules in that ovary, sure, but still, single ovary. Here, it gets more complicated in the aggregate fruit. The name also gives you a clue that you're putting more things together, okay? Multiple units. And here, one flower, but many ovaries. So it's an aggregate of many stigmas have been pollinated within that individual flower, and many ovaries are being fertilized. So this stays the same, still one flower, and this is greater than or equal to 2, right? AKA many. And then the next one, in the multiple case, both things change. You got now many flowers. You got the pineapple, right? Each kind of diamond unit here was a flower. And an ovary. So here it's like simple, 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 right? All put together. So this is greater than equal to 2, greater than equal to 2. Here, in the aggregate case, it's still just one flower, but it has lots of individual ovaries put together. This one, each flower has one, and those are all stuck together.
They're all different, okay. That's what hurts my brain sometimes. I just think of it. That's all like fused together into like one fruit, basically, or like how does the.
Yeah, and how that looks varies, you know. But it has to be some kind of unit that's like, makes sense for seed dispersal. Either it's a consumable thing that then something can move out, or it can drop it, or it can knock over, or it can roll somewhere. But if it was like separate various things, then each one would have to have its own seed, or it wouldn't make any sense.
Sure.
You know, and that happens, right? There's simple fruits that have lots of fruits on a plant, and they just, each one drops, or each one gets eaten by a bird, or whatever.
Sure, it's a different strategy.
It's a new topic, but I've been kind of hooked up on like the Krebs cycle, and is it the same thing as a citric acid cycle?
Yep, 2 names for that
And then
thing.
it says, where does it take place?
place, you can also frame that question as what organelle does it take place in? And then you want to think about what's the overall, what's the big result of this whole process that Krebs is part of? What'd you get? What's that? Mitochondria. So what you get is energy, right? The thing that makes energy is the mitochondria.
I think on the quiz it said, explain the Krebs, what happens in the Krebs cycle, and I couldn't answer it, so I figured.
Yeah,
might as well
right.
go with credit.
That was an open-ended one
Yeah.
to fill in your understanding. So some things that happen.
Quick question on that too.
Yeah.
Just regarding respiration. The first step of respiration doesn't take place in mitochondria, right? It's in the cytoplasm.
Correct.
Okay.
I'm not gonna split hairs on that, because this isn't a cellular biology class. But what I want you to get the connection between respiration makes energy, mitochondria is the place that produces it. So that first step does take place in the cytoplasm, and it's moving it across the mitochondrial membrane from the outside into this thing that then allows that first little bit of ATP to get produced. So it's like when you ram things across the membrane, it's good chance to tack stuff on. But then that big payoff from the electron transport is from the mitochondria. And so, Kayla, to come back to that question, it's the breakdown of acetyl coenzyme A. We're taking glucose, right, the answer to all these questions, and making it into these smaller carbon molecules, the reverse of photosynthesis, get a little bit of energy there, and carbon dioxide is released in that step.
Can we just go over respiration? Because I think the electron transport part really confuses me too.
For the purpose of this class, the things to know about electron transport are that it's in respiration, right? And that respiration is the process that makes energy. That it takes oxygen, that it's again, the fuel for the energy fire is the oxygen. It's like you blow on that fire, right, a real fire,
Yeah.
right, and it gets bigger. You blow oxygen into this process, and you get more energy out. So this is respiration, it's an energy-making process. In the presence of oxygen, you get a lot of energy, and that happens in the mitochondria. And that a bunch of that's lost as heat. If those points make sense, then you're gonna get all those questions right. I mean, that's the base of that process, right? We're taking glucose, sugar, we're ripping it apart. Those bonds that are getting ripped are the energy that we're using. And as part of that process, gives off water and carbon dioxide. Opposite of photosynthesis, where water and carbon dioxide go into the process. And remember, that's connected to when respiration is high, that means temperature is high. Water is being released every time a mitochondria rips one glucose apart to make energy. Water is being released. So, at high temperature, high respiration rate, a lot of water loss. And there's going to be additional stress because of those temperature factors. That's going to mean, right, it's evaporating from the soil. This plant is under pressure to absorb a lot of water to keep this process going. At the same time, it's going to want to close stomata because it's hot, right, and not lose all that water. And so it's like, it needs to get the water out. It has to release all this water, but it's getting hot and starting to suffer. The stomata are closed, so now it's getting hot inside, and that's when plants suffer. That's when you see real damage.
Like, I don't need to like go into like complete detail about it.
No, I'm not gonna ask you what happens to FADH2 or anything.
Yeah.
Keep it to the core purpose of the thing.
I was watching those videos that were posted on the equipment, and I was like, I feel like explain what happens to TH and all that, like after it like crosses over the
No.
membrane and all that.
I feel like I've been pretty clear about that.
Okay, that's all I make sure.
Just keep it to the core information. What goes into respiration overall And what comes out? Remember, the most important thing that comes out, a bunch of energy. On your little plant life, or our larger mammalian life, just like what goes into photosynthesis, is a lot of energy that comes from light source. And the rest of these things are the same components shifted around. This one's building things up with some raw ingredients that exist, right, in the atmosphere, in the soil, making a thing. Here, we're taking the thing in the presence of this igniter, right, and making energy, and in the process, these are released, carbon dioxide and water. So, again, if you know one of these, you know the other one. And if you know one of these, then you can derive where these things go. And that connects to that concept of a limiting factor, right? Which is just like, hey, if we don't have enough CO2 in the environment for photosynthesis, it stops. And so now we're not making food. And so how do we get more CO2 from the environment? Open those stomata. And again, it could be a challenge to balance the environmental conditions with that need for, right, one of the 3 ingredients in the recipe for dinner, right, making that glucose. Yeah.
A question on hormones and tropism.
Okay, yeah, let's see if we are done here. Go back to that.
We go into the stuff like all of this.
Sure. Let me finish mentioning these two. So again, I put together some comparisons. This one I think especially, right? One's building, one's breaking down. One produces water, one uses water. Like if we can see that and it makes sense, or if you can look at one side and fill in the blank on the other side because of the relationship between those, then you've got the necessary material. In terms of glycolysis, again here, let's not get lost in the weeds. This is first step in respiration. What's important is you need energy to start, right? Just like everything. Photosynthesis needs that solar energy to start. The light reaction, the light independent reaction, or Calvin cycle, needs the ATP that's made in the first step. Here, we need a little bit of that to get going. And we're breaking down glucose. That's it.
That's good.
That's it.
It's just getting that too. And the pyruvate, and then the pyruvate starts the Krebs cycle. Okay.
And yeah, so related questions. You know, Kayla, when you're just writing glucose for everything, you can say glucose, and then here is broken down. Okay? So maybe it is the answer to everything, part of the answer to everything. Andrew.
Just curious about final review question 51. It says, if you want to speed up the plant life cycle, which hormone should you use? And so like some of the hormones are growth, some are inhibitors. And so like one might speed up the beginning of the life cycle, but the ethylene might speed up at the end. So I was curious what.
You're right, that's not that specific of a question.
If I would add, yes, I would say ethylene, just because it starts the process of senescence and survival.
Yep. So that's the overall process racing toward senescence and death, right? If I said, which one speeds up flowering, then you could tell me it was ethylene or it was gibberellin. If I said, which one speeds up shoot growth, what would it be? Auxin inhibits shoot growth, right? That's what apical dominance does. And it encourages reproduction. It's that it's the rooting hormone. Yeah, definitely keep that one oxen cytokinin, that relationship, one inhibits the other, oxen roots, cytokinin shoots, and then it stops that from happening in the other one. Have that definitely in your back pocket.
I have another question about photojournalism. So I know, like, they'll grow towards the light because oxygen likes to stay out of the, like, away from the light. So is cytokinins, like, gathering on the part that's growing, or like, how does that balance out? Like, is there,
Well,
like, more
cytokinins primarily produced in roots,
Yeah.
so there's not much of it up here to migrate around. So because the apical bud, that meristem, is the site of oxygen production, that's why we see most of this reaction. No? So it's getting made here,
Yeah.
right? And so we see the reaction to the light there. The cytokinin's only there if it's transported for a specific purpose. Like it's going to make
I see.
those buds here grow, so it's gonna move it up, right? But it's not always just sitting there exposed to light at the top of the plant.
I get it, okay.
Yeah.
Let's see, is number 53, in a greenhouse setting, why might long night plants need some type of cover? Is it to like mimic that?
Mimic what?
The light cycle?
It is, but be a little more specific. Long night plant, AKA short day plant, is this side. And so, cover, what would be the purpose of cover?
Make it seem like night?
Huh?
Make it seem like night, or?
Make it seem like night, to keep that night long, right? This is the poinsettia example without directly calling it that. So for poinsettia, short day, long night plants, post autumnal equinox, the nights are short, right? And it's getting that signal to flower. If we want to delay that so that our Thanksgiving poinsettia sale, right, looks, right, all eyes are on those red bracts and not on the flowers, right, steal the show. I mean, they're not that showy and they're small, right? But, then we need to reduce the signaling to flower, and that would be by blacking it out, right? Night long. In the opposite scenario, something we wanted to flower for marketability, right, then we would remove that so that we could shorten up the night, or we could flash.
I have a question about one of the inhibitors,
Abscisic acid.
so it inhibits seed germination. Now, is that only when it's still attached to the plant, or is it also there when it's off and then it waits for gibberellin to activate?
Because they're both in the seed.
Okay, so like even when they're not on the plant, it's still,
That is still a cell that can produce them, right? That seed needs meristematic tissue, just like the rest of the plant cells. Yeah, seed, no, okay. No, doesn't see that.
so the acid is what's holding it off until it has that prime real estate.
Yes, is one of the signals there.
Okay.
So it responds to that inhibition being soaked in water as an indication that conditions are probably okay. And then that chemical signal starts the actual development process. So it's like, it's not the only thing, but it's part of the cascade that makes it happen. So, seeds, and remember, importantly, the radical pops out first. You see root hairs, right? And if you close up just like intestines that are folded up for surface area. Root hairs, here's that big root, and then all these hairs. Sorry, there's probably a better analogy than intestines. Right, but each one of these, you got all this and all this, this and this and this and this, right? That's all additional surface area for water absorption. If it's just that, right, it's not so much. So now we've got way more opportunity to do that. Maybe more like the microvilli in our lungs, rather, all folded up to get that oxygen. Of course, we got ways to break dormancy, and like we just said, water division is the primary to getting the rest of the signaling happen through other examples. Two different ways of germination, depends on where the cotyledon is at that time, below or above the ground. A little bit of a difference in terms of risk initially at germination. Your cotyledon's above ground, could be chopped, could be frozen, could be chewed on, and then you lose your food source because this little guy doesn't have any true leaves yet. Right? So that's all it's got for food. So protecting that thing can pay off. Or the hypogeel germination. Look at that. Of course, temperature is connected to germination with an optimum temp and a range of temps. And then my point about no true leaves. And this guy's losing mass because it's eating the cotyledon or endosperm food source until the time that it can photosynthesize enough to start building back up. And of course, that photosynthesis is creating glucose that's going to get used in respiration to extract the energy to grow that seed. What else?
Could you answer 46?
You gotta tell me what it is.
Which type of plants are most prone to respiration?
Which type of plants are,
Most prone to respiration.
I can't answer that. Anybody else?
C3.
C3 plants, yes. And why?
Folks in Montana are always a little bit open.
Yes, sir. So,
Stomata, C3 are always a little bit open.
so that's C4. C3 is opening and closing, responding to the temperature situation in the water demand, and that makes the problem.
It's mainly just because they're using Profister, right? Because that's the enzyme that's binding to oxygen.
Yeah, exactly. That's the one that will also bond oxygen, that creates photorespiration, right? It doesn't pay off with glucose, doesn't pay off with energy. And C4 plant with stomata always slightly open, that's the workaround for avoiding this situation, because then there's always air coming in, which contains that very small percentage of carbon dioxide that can be used.
The C4 shuts the stomata in response to the heat or whatever, and then oxygen builds up so much that it's more likely to photorespirate.
C3, yes. So it's hot out, C3 plant, closed stomata, now new CO2 is not coming in, all the internal CO2 is consumed, this enzyme starts grabbing on to oxygen. Instead, it'll preferentially take CO2, but if there's none available, it'll settle for oxygen, but then you don't get, right, you don't get that food result, that glucose, or energy production at the end. So it's not a good move, and thus, we gotta open those stomata again to get more CO2 in to deal with that, and of course, it's hot out, so now we're losing water at the same time. With C4 plants, always slightly open means CO2 is accessible all the time. And likewise, can plants, crestage acid metabolism, are closed during the day when the water loss would be the highest in these hot conditions. And then they store that CO2 from opening at night as this alternative compound malate. And then during the day, when they have sunlight, they do the first step, the light reaction, right? Change that stored thing back to CO2 that they can use in the Calvin cycle of the light reaction so that they can grow. And all that changing around to get around stomata closed all day takes extra energy and slows that growth down. But very effective. Slow and steady, won that race, for sure. So yeah, keeping those two straight, CAM and C4. Okay, done a little bit of comparing for you. Like slightly different strategy. Position of the stomata is different. Open a little bit all the time. Open only at night. C4 moves it in space. Right? That bundle sheath. CAM moves it in time. And they both use PEP instead of Rubisco. That does not bond to oxygen, avoiding photorespiration, which I mentioned here. It reduces growth, right? It could be 20% less growth when you're hot and photorespirating often instead of opening stomata for these C3 plants. Instead, CAM and C4 plants don't have that risk with PEP instead of Rubisco, so it's more efficient. If they didn't have that option to avoid it with this different enzyme, the already slow growth, especially for the CAM plants, would be even slower, right? Reduce that by 20% when temperature is high and humidity is low, and you're gonna see plant size in that desert reduced. You're going to see less reproduction because overall there'll be less food and less energy. So, try to see beyond answering my test question, the relationship with things like if stomata are closed, then as you work backwards, that means we're running out of CO2. That means water is not getting out. Like, what are the effects of that? There's a story about water there. There's a story about carbon dioxide and photosynthesis there. There's a story about respiration there. And they all come together at that spot. That's the threshold between inside the plant and out. Caleb?
On the topic of respiration, number 37, it says, what is the equation? Already got that, but where does it happen? Is it in the leaves or is it also just like mitochondria as well? It seems like that was in the stomata, like.
So,
Where does
leaves are a
respiration
plant
as a whole-
organ, and stomata are in the leaf, and mitochondria are in all the cells. So it could be all of those places. The answer is every living cell. Every living cell. So that's respiration. That is what living is. Making and using energy. If you can't do that, as far as we know, you're not living. So all those cells have mitochondria because they need energy. Prakash,
You kind of answered it already, but I just want to make sure for the question you were just talking about the C3 plants and the Rubisco.
Okay.
So, which type of plants are most prone to photorespiration and why the C3 plants because of Rubisco and lack of environmental adaptation? As like a roundabout way to get to that connection that you were talking about.
So Rubisco, yes, and this is a great point generally. Lack of environmental adaptation. If you write that on the test, I can intuit the answer because I know the answer, but you didn't say any specific terms that indicate
Like this.
that you know that it's because the stomata are closed and CO2 is not in there, okay? So for any question, you want to be specific. And being general, my assumption is that you don't know, and you're trying to be vague so that I fill in the blank. But be direct. Like, when I go for a question, I have like, if that question is worth 10 points, I say, oh, there's 5 points if you tell me about Rubisco, and there's 5 points if you tell me about the stomatal position. And if you don't do that part, right, I don't just be like, Akasha loses 3 and that person loses 6. I said, no, that's what that's worth, and you didn't say it. So try to, cut to the chase.
Explain what the environmental.
Yeah. Yeah.
Please correct, my first assumption here is respiration, aerobic respiration, only 40% is kept, right? 60% is lost to sea. Is that true for anaerobic, or is there just a different ratio and don't worry about it?
It's, for the purposes of the exam, don't worry about it. But it's different because a lot of that heat is generated in the electron transport where, right, like 90% of that energy comes from. And so anaerobic does look different in that the percentage of heat lost is less, and likewise the amount of energy produced is less. And that's, that's fortunate for some products like we can ferment a lager, right, at cold temperature. That's, a lager yeast is meant to be fermented at 40 degrees Fahrenheit, and you get a result. Whereas if you put other organisms at 40, metabolism is slowing way down. What else? Roots, stems, yeah.
Oh, I guess we could go with stems, but,
I'm just saying.
what's the thigmotropic response? I think I missed the,
Phototropic response.
the thigmotropic.
Thigmo, oh yeah.
Clingy,
What's that?
the clinging plant.
Clingy.
Oh, okay, got it.
Thigmo, think touch, contact.
Okay,
And
so like binding and lumbar tendrils.
Yeah, exactly.
Okay.
And that's all you
Okay.
need to know about that, right? Is that a positive thigmotropic response is like bind onto that thing, grab onto it. And a negative would be a root, like avoid that rock, because that rock's not going to hold any water, doesn't have nutrition, it's going to limit the root growth. So it's advantageous to move around it, and maybe there's a good water source below, or maybe it's in a boulder pile and there's nothing else to do, but at least it's trying then to find the next option. What else? Roots, stems, leaves, flowers and fruit, seed germination, photosynthesis, respiration, hormones, temperature, stress, plant nutrition, and seed germination again. Temperature, stress, we didn't talk about it all yet. Let's take a quick look. Chilling and freezing, right? There's a difference in temperature there. One's above freezing but cold. One is below freezing. And the difference matters because that's where water freezes, right? Plants are filled with water, and water expands into solid form of ice when it's frozen, and that's going to explode cells, or if there's a lot of water between cells, it could also damage, right, the outside structure. And so plants got two basic strategies: avoid something, right, or tolerate it. Learn to live with the colder temp. So they can tolerate it by lowering that freezing point internally by packing salts in, right, the equivalent of rock salt on the sidewalk, and they can avoid it by moving one is get water in with lots of salt to lower the temperature of freezing, and that's useful to a point, right? That's gonna be more useful at 28 than it is at 0. Once you get there, you need to start moving that water out in hopes that it will only expand in between the living portions and not explode the cells from the inside out. And even if it does kill some cells on the outside, hopefully it doesn't kill them all, right? Maybe the leaves that are most exposed get damaged because not only do they have internal ice, they're also where the wind is coming through, that's drying things out faster, making it colder, so they'll get more damage, but maybe some of the leaves that are closer to the stem and a little bit protected from the other foliage survive, and then you can keep going. As long as you got, functional roots and some meristematic tissue, right? A couple of nodes somewhere, then forge ahead, build it back up as soon as conditions are decent. The heat stress in both of these. The tricky part is the effects are often the same. Wilting if it's hot, wilting if it's cold, wilting if it's over water, wilting if it's underwater. So I'm not gonna just how do you look at a photo and tell me what that is? But I will want to know how do plants respond to temperature? What's their strategy? And anything else?
So with heat rate of more respiration, less photosynthesis, that's where you end up with photorespiration. Or that's a tangential thing.
Both can be happening at the same time, but it's not all interrelated. So respiration rate is just a factor of temperature. Photosynthesis rate is a factor of temperature as it falls, but also involves the presence of water, carbon dioxide. So let's do that there. So we're talking about, in this case, very high temperatures. Respiration rate is still high. No photosynthesis, so consuming any stored resource, starting to run low. Giving off water at the same time. And then, what's the other piece? Respiration, photosynthesis, and maybe you just frame the question again.
Oh, I guess what is happening to the, photorespiration. Photorespiration is also happening because it is hot. and
Oh,
it's
yes,
water
yeah,
close,
so
or not.
in C3 plants,
Okay.
anywhere up here, pretty much once you're past the optimal photosynthesis temp for that plant, photorespiration, as it gets too hot, is getting increasingly likely, because as the heat increases, that plant's gonna try to close the lava and save its water resources. At the same time, it has to get this water out that's being produced more rapidly now in respiration, and so it's not only not making energy through photorespiration, but it's losing energy through regular respiration at the same time, getting itself into a negative feedback loop of running out of energy and burning more energy to do that.
So, but respiration requires the stomata be open to release water.
Not immediately,
Okay.
but because that water and carbon dioxide are released, as temperature rises, if there's a lot of water vapor inside the leaf that's heating up,
Yeah.
that's gonna result in heat stress damage to the tissue if it's not released.
Got it.
And so when it gets released, now the plant's losing water, and it's gonna need more, right, from the root zone somewhere to keep that whole process going. At the same time, it's burning up food stores, because only respiration is happening here. photo restoration is also happening. So recognize that photo restoration and restoration are not the same, right? Photo restoration is like an aberration of photosynthesis, where instead of glucose, we're getting CO2 and no food. So that's happening simultaneously, and we're burning up all of our food store, and we're not making any more. So things are getting worse, right? And that's where we see, right, a lot of damage. I mean, this is really high temperature. You know, if you're up there, your plants are suffering all the time. When you're in, you know, the 90 degrees range, then the average C3 plant is starting to suffer, but it can cope if the water resource is adequate. It can keep stomata open, keep burning through water as it respires, but cooling itself off. Okay? That water holds a lot of heat, and so by getting it out of the plant, you're cooling it, and that's keeping it good. But as you get hotter and hotter, the risk just increases.
Can we go over how like salts build up in the cells, like facilitate osmosis and all that?
Yeah. So, there'll be some question about these. One active transport, two passive modes. One's diffusion, right? That's the osmosis of water. The other's facilitated diffusion, which is with membrane-bound protein. Both of those don't require energy because this higher concentration to low gradient exists. Right? The opposite situation here, you got to put energy in if you want to move something, you know, against the grain. So that's the basic setup for this. And then,
So it would just build up salts, like via membrane, and then since there's a higher concentration inside of the water, in
yep,
or outside.
yes, inside. Helpful to look at it with the water and the solution, right? The salts or whatever's in there. So, like you're saying, by packing more salts inside the vacuoles of those root hair cells, which are connected to the outside, right? That's the epidermis.
Right.
Water wants to move from where it's in higher concentration to lower. Because the inside, the balance is some salts and some water. Outside, it's maybe all water, or mostly water and a little bit of salts.
Yeah.
So water's moving in to dilute out that salt solution. From a higher concentration of water to a lower concentration of water, right? Dilute that out, and in doing so, moving water in, absorbing into the root without spending energy on it. It's the same concept that fills the vacuole in the guard cells to become turgid and thus open the stomata. And in both cases, you're manipulating the gradient by putting, go ahead. Harrison. By putting more salts inside so that water flows toward that, to dilute it out, to make an even, balanced solution. I mean, that's how the physics works out. It doesn't always end with balance, but it's going towards that. What else? Well, if that's it, we can end it there. If you have questions this week as you're studying, by all means, come see me, send me an email, get that answered. Oh, wait.
I have a question I have curiosity. I don't think you're gonna ask on this, but like, what during photo restoration, like what happens after the oxygen is binded
Yeah, I was like, is it just waste?
But, so, in a rough sense, you're saying what happens here.
Yeah.
So, energy goes into this. Instead of building something up, stacking those carbons together, that part doesn't happen. So that carbon ends up getting released as carbon dioxide instead of going into glucose. So that carbon goes away. Now it's released, and it took energy to get there. So that energy that was produced from that first sunlight makes a little bit of energy that could have powered the Calvin cycle. Now that goes here too. So you lost energy, you lost glucose, and you released the thing that you need for photosynthesis. Yeah, you just, it's the opposite of 777, where you lost every which way. Gus?
So we were just discussing the nutrition is not on that, and so we were wondering how much of that subject to be on the.
Yeah, good point. I didn't get to the nutrition last year, so we didn't have that on there.
And nobody then wrote any questions about it. But as far as it goes for nutrition, important things are, I already talked about new leaves, old leaves, mobile and immobile nutrients. That's a definite. What's EC? Electrical conductivity, which is a measure of total salts in the solution, which tells you something about nutrient volume, right, amount, but not specifically you're low in phosphorus. But that, coupled with pH, you should also know, tells you both do you have nutrients and are they available to the plant. So they're two separate things. Your soil solution or your soil-less media can have nutrition, but it's not necessarily getting to the plants, and having an appropriate pH facilitates that. So I'm not going to ask anything like, tell me where iron is most available, but the idea that there's a range where nutrients are more available and a range that they become less available is something I want to see if you know. The relationship between pH and how the plant can get nutrients. Mr. Good.
They always leave aluminum off of this one, what is the deal?
What's that?
They always leave aluminum off of this chart, it's on there, it's on there. And then like that leads to like toxicity once you get down into the acidic pH range, aluminum toxicity. And,
Yeah.
I don't know what,
Aluminum toxicity is a real thing, but aluminum is not an essential nutrient,
yeah,
right? So this is
that's why
just,
they excluded from this.
yeah, this is just the plant, you know, and even here, this isn't all 17, well, 14, I guess, without the carbon, hydrogen, and oxygen. But, you know, there's over 120 elements, and it's not meant to represent all those, just the ones that plants need in the most amounts. But yeah, there's aluminum in soil, and it's harmful, the same as like arsenic or something, but they're not, there's no deficiency because they're not required.
You're just saying as long as we understand the relationship between soil pH and availability of
Yeah.
pH.
Yep, that's it. There's a range of pH where nutrients become unavailable, and thus you want to be in the available range to facilitate uptake. And one, besides doing a pH test, another way to get information about where you're at is EC testing, or like, what's the total available nutrition here? Again, it doesn't tell you how much of each thing, but it gives you a picture of the total.
But we need to know about, any deficiency information at all, just what you, or just what you said.
You'll need to know that deficiency symptoms show up in the new leaves on immobile nutrients and in the older leaves first on mobile, and why. But yeah, I'm not going to be a full part of the class, but I don't have the time left to like provide samples and say identify these deficiencies. Right? Did Dan do that? that in soils? No? Okay. You used to do that. So yeah, I'm not gonna like give you one of these photos and say, tell me which one, but I will try to get at this information. Conveniently for plants, NPK, magnesium, all macronutrients needed in large amount are mobile, so that's useful. And that mobile means it's taken from one spot in the plant and used somewhere else during development. Instead of, just to circle right back to the beginning, the immobile ones are stuck in place at the site where they're first used. And so there's no recycling within the plant. Eventually they'll break down, be used again elsewhere, but they stay put and thus can't be repurposed down the line.
This, I was wondering, this in the beginning of the class, it might not be critical, but this is related to like what the phloem actually can transport, right? Because you were saying earlier that a lot of these minerals are flowing upwards in the xylem, because that's where they're dissolved. But then, does that mean, like what can move down, if anything? What is actually moved in the phloem? Is it mostly carbohydrate?
Kayla?
Glucose?
Yeah.
Full circle. It is the
Sugars, hormones. And some nutrients can be moved, but it's a combination of size, structure, and where those things are used. Like if the calcium is incorporated into cell wall, it's not going to then take that cell wall apart and move it somewhere else. Only available from things that would not be death for the spot where it's using. And it's hard to get them there, or sometimes impossible. And phloem's mostly does great stuff.
This is not on this topic, but it is on the photorespiration. So we were talking, we were curious, like what's the point of photorespiration if it's like not really doing anything beneficial to the plant necessarily, like not creating energy? Like what is the point?
What's the point, man? Anybody got a thought about that?
I didn't think of it as like an intentional step in the plant's process, but
It's not.
it's just what happens when you're using Visco.
Yeah, there's no point. It's just an unfortunate
Thing that happens. Okay.
result of that, you know, the evolution of an enzyme that will grab carbon and make food out of it was a big deal. The fact that it had this downside, right?
It's just like that.
It's better than nothing.
Yeah.
Or you gotta come up with another one. There's only 2 of them out there, so it's not that easy to come up with, I think. So you gotta take what you can get when you're a plant, you know?
Okay.
Yeah.
So
It's
it's
just-
just a process that happens when these conditions are existing.
And, you know, when this started, you know, early plants 350 million years ago, there was more carbon. This is just volcanic eruption, right? Ash-laden atmosphere, and maybe it was less likely, and thus less damaging. And then things changed. Now they're going back up, so maybe someday we won't have this again. It's not what I'm trying to say. What else?
So C3 plants, like, are the older plants, right? They came before like C4 and Camp. That was, kind of answered my question.
Yep, they're like the old model, it's still like manual doors and locks, you know. Not perfect, but does the job, you know, and then sometimes you look back and you're like, you know, I kinda like that. I promised at the beginning is there now, okay, all the way up and out.
There was another topic I forgot to bring up, it's the one that like doesn't need to be fertilized. What was that one called?
Parthenocarpic.
Parthenocarpic, that's right, yeah. And that's, so that one, it can be fertilized, but it doesn't have to, or does it just never have to be fertilized, ever just produces that way? Like, does it produce a flower still, right?
Yes,
Okay.
but it doesn't have to be pollinated or fertilized. It could be,
Yeah.
right? But if it's fertilized, right, then we don't. But it could be pollinated but not fertilized and still get a fruit. It could be not pollinated and still get a fruit.
Got it.
Yeah, that fruit's gonna get produced one way or the other, as long as the flower is able to develop. Of course, there's no additional genetic material, right? So that's a clone,
Oh, okay.
Right.
Yeah. Yeah, makes sense.
there's no mixing.
Yeah.
That's all one organism.
Yeah.
And that creates that banana disease situation that I mentioned, where 98% of global bananas are one variety. And so we hit a disease that we can't manage well, and it's going to wipe out the population of that. We'll have to come up with another variety that is, resistant to that pathogen.
So, okay, so if it can clone itself that way without a sperm cell, like fertilize it, like how does that work? Like how it's not only that how you clone itself, it doesn't, why not? But yeah, that's exactly how it clones itself. Okay, no.
Well,
I gotta work out on it.
So the seeds
Yeah.
in that banana, that parthenocarpic banana, are not viable.
Right, yeah.
Right, because there hasn't been fertilization.
Yeah.
Right? So genetically, it's the same as the parent plant. But how that develops is a long story.
Okay.
All right, well, thanks very much. Again, I'm here to help answer questions until Monday, bring those great assignments. Student assistant application to work with us in the winter, get tuition covered, partially covered, is due next Tuesday at